and welcome all. Myself, Dr. Swapna Kumar Srivastava, Associate Professor in School of Biotechnology, IFTM University. On behalf of my university, I would like to welcome all the participants attending international webinar on Yeast contribution to Alzheimer disease. IFTM University is one of the topmost university committed to provide an environment for skill development and knowledge building by emphasizing on teaching, engaging in research, and participating in various community activities and collaborating with national and international repute. On the bottom of my heart, I would like to thank our Honorable Chancellor Sir Shri Rajiv Puriwal, Co-Chancellor Sir Shri Abhinav Puriwal, Honorable Vice Chancellor Sir Professor M.P. Pandey, Registrar Sir Professor Sanjeev Agrawal, and Co-Vice Chancellor Sir Professor Rahul Mishra for providing such a skill-based platform. Information is knowledge. Knowledge is wisdom. The only source of knowledge is experience. You need experience to gain wisdom. All these are well suited to our speaker, Sir Geoffrey, Honorary Professor, RMIT University, Melbourne, Australia. Let me introduce his brief profile. Dr. Macreadie, a PhD in Yeast Genetics and Molecular Biology from Monash University and a postdoctor from University of Texas Health Science Center at Dallas, and played key roles in identifying the structure and function of mitochondrial genes, encoding 3 ATPs subunit, the ribosomal protein VAR1 and endonuclease encoded within a mitochondrial intron. During his 25 years long career at CSIRO, Division of Protein Chemistry, Parkville, Dr. Macreadie developed highly versatile novel yeast expression systems that were marketed internationally by companies including AMRD and Biotech. Inspired by the success of the hepatitis B vaccine, Dr. Macreadie's first project at CSIRO, one of the first projects funded by the National Biotechnology Program and led by Ahmedabad, aimed to produce a subunit vaccine for infection brucell disease virus, a major disease of poultry. The yeast expression system enabled production of successful vaccine, the world's second viral subunit vaccine. From 1990 to 2000, Dr. Macreadie seconded to Biomolecular Research Institute led by Peter Coleman, where he continued to the use of yeast expression system and employed them for the study of HIV/AIDS protein, VPR, NEP, PPU, and VIF. Later at the Biomolecular Research Institute, he focused on sulfur drugs resistant and took a particular interest in the folate synthesis pathway, which are the target of the oldest. Chemically synthesized drugs, the sulfur drugs, with Peter LKS, he resolved engineering yeast to show that DHPS mutation associated with sulfur drugs in AIDS association pathogen pneumocytosis, Geoffrey, did indeed cause drug resistance in yeast. These sulfur drugs are textbook example of the competitive inhibitor of PABA, but with Onisha tail, he showed that they could be conjugated in the place of PABA to form sulfur dihydropeptide and to inhibit DHR as well. With Mike Lawrence and James Swarbrick, he went on the screen for new antifolate for malaria and pneumocystis based on the structure studies of DHPS and HPPK. More recently, he was focused on development of yeast model for Alzheimer disease to aim to aid in the understanding of chemo preventative for Alzheimer disease. From 2010 to 2020, he coordinated courses on industrial microbiology and protein technology at RMIT University. While continuing research on chemoprevention of Alzheimer's disease, exploitation of microbes for their commercial benefit, and understanding the requirement that enables successful microbial culture as well as their preservation. With 176 research publications and 6 patents, to his credit, Dr. Macreadie has awarded with 1990 Fulbright Senior Scholarship Award, 1996 Frank Fenner Research Award, 1997 CSIRO Chairman Medal, 1998 Macreadie Traveler Award, 2002 Respold Robertson Research Award, 2004 EMBO Short Term Fellowship, 2005 Merck Sharp and Domi Mycology Award, third place in Bioscience Technology Research of the Year Award in the Australian Society for Biochemistry and Molecular Biology from 1997 to 2000, and is the Editor-in-Chief of Microbiology Australia since 2006. He was the chair of the organizing committee for the 23rd International Conference of Yeast Genetics and Molecular Biology 2007 and the 11th International Symposium on the Genetics of Industrial Microorganisms 2010. I am Joe. Today we are very benefited with the expert lecture of Macreadie. Now I will invite sir. Sir, please bless us with your valuable knowledge and your experience lecture.
Thank you. Thank you for that kind introduction. Is someone going to start? Am I going to show the slides or is someone going to present them? Would you like me to share? I can share my screen, I think.
So the screen is not visible.
Okay, so I won't see you at the moment, but I'll go through my presentation. So this is Teachers' Day, which I've done a little bit of research on, seeing that you're celebrating this in honour of the birthday of your second president, and it's good to be here and presenting to you. My talk's on the yeast contributions to Alzheimer's disease, and I wanted to start off by just acknowledging the importance of not only teachers but students, and these are some of the students on the left column who have contributed to this work in these studies of A-beta at RMIT University. Several of them have come from India and have got themselves well established in their careers now. Some have come from other places as well, and I'd also like to acknowledge the teachers on the right-hand column, Paul Ramsland and Benu Adhikari, some from CSIRO where I previously worked, and I don't know whether Ashok Dubey is on the line, but he worked at CSIRO as a vacation visitor and contributed to these studies. Also, my son started off, I guess, doing some studies with me, and he's now a professor at Deakin University. So good students can become, hopefully, good teachers, and I just want to encourage you to pursue excellence in your studies. So just to talk about Alzheimer's disease now, this is a disease of ageing in Australia. 10% of the population above 65 has Alzheimer's disease, and as you get older, the frequency increases at 30% above 85. So what causes Alzheimer's disease? These are some of the things I'll discuss. Can we find compounds that prevent it? How can we overcome it? So it was actually discovered and first reported back in 1906 by Alois Alzheimer in Germany, and one of the people that was suffering from it was Auguste Deter, and she died, I think, at age 55. She didn't know her name. She didn't recognise simple things in daily life. She had an early onset version of Alzheimer's disease, and it's a sad disease. Particularly, I guess those that have it perhaps don't notice it so much, but for those that are caring, it's a real strain as people suffer from it and eventually die from it. So I think it's a disease of proteostasis. So proteostasis is a term that refers to keeping proteins in a balance. And as we age, proteostasis declines and some proteins accumulate, including some that are toxic. You can liken it to perhaps just having a mess in your life or accumulating rubbish. If you don't clean it up, it's going to cause you a problem, and that's, I think, what some proteins can do. And the particular one that's associated with Alzheimer's disease is this A-beta, amyloid beta. It's derived from a natural cellular protein, and you can see here the two proteases that cleave it from this precursor protein, which is Alzheimer's precursor protein. And it's a 42 amino acid protein, not modified in any way. It accumulates with age, and it's found in the plaques in brains, and that's one of the reasons why it's been particularly linked with Alzheimer's disease. It actually turns out to be toxic too. So our studies were to see if we can find out more about this A-beta. Where it comes from, this just shows portion of APP, the precursor protein, and the A-beta is this part in between, and you can see the sites of cleavage of the beta-secretase protease, which is a prenylated protein, and gamma-secretase. And this is some of the evidence for genetic evidence that A-beta is a cause of Alzheimer's disease. These are mutations that are associated with early onset of Alzheimer's disease, mutations in this or around the sequence that leads to the cleavage and production of the amyloid beta. So that leads to early onset Alzheimer's disease, but Alzheimer's disease is something that can affect anyone. It's this one, these mutations cause the early onset, but with aging, that wild-type version accumulates and causes problems as well. You can see an example here of how much it accumulates in age. So by the time of well-recognised physical appearance of Alzheimer's disease, you've probably got about 6.5 milligrams of A-beta in your brain. So it's accumulating constantly. Another study that implicates A-beta is involved in Alzheimer's disease is in Iceland. They do whole genome studies. They've got good information about the genomes of their population, and they've actually found that there's a mutation that's protective, and it's published in Nature. The reason why it's protective is it leads to much, much less A-beta. So another bit of evidence that A-beta is very important in the causation of Alzheimer's disease. Now, just to give you a little bit of background on yeast, I've been working with yeast for my professional life, and it's important because it's a eukaryote, just like us. It's got mitochondria and a defined nucleus and organelles. So it is genes in yeast can be likened to many of the genes that we have. It's been possible to transform it since about 1978. It's non-pathogenic, it's easy to culture. The genetics and molecular biology is well understood. We've had the total sequence of the yeast genome since 1996. And it's got an excellent track record in biotechnology, and you can see here products, beer and wine and bread, the traditional products, but also some of the novel products, genome studies. They've got good information about the genomes of their population, and they've actually found that there's a mutation that's protective, and it's published in Nature. The reason why it's protective is it leads to much, much less A-beta. So another bit of evidence that A-beta is very important in the causation of Alzheimer's disease. Now, just to give you a little bit of background on yeast, I've been working with yeast all my professional life, and it's important because it's a eukaryote, just like us. It's got mitochondria and a defined nucleus and organelles. So it is genes in yeast can be likened to many of the genes that we have. It's been possible to transform it since about 1978. It's non-pathogenic, it's easy to culture. The genetics and molecular biology is well understood. We've had the total sequence of the yeast genome since 1996. And it's got an excellent track record in biotechnology, and you can see here products, beer and wine and bread, the traditional products, but also some of the novel products. Yeast is made to produce hepatitis B vaccine, which I think we've all had, Gardasil, which the girls will have had to protect against human papillomavirus, and this one protects against hepatitis B virus, and these are leading causes of liver cancer or cervical cancer. And of course there's insulin too, so without the yeast we wouldn't have people's lives being saved if they've got type 1 diabetes. So it's also possible to secrete the protein, and most importantly, yeast is a safe organism. It's been used as a model for human health research, including, these are some of the things that I've used it for. I've used it to study AIDS and the auxiliary proteins of AIDS, to study antimicrobial resistance, to study the actions of drugs, and neurobiology, and it's also a model for aging, for human aging, and I'll come back to that shortly. It's easy to manipulate yeast, it grows rapidly, it's cheap, it's generally regarded as safe. You can do good genetics with yeast, it's got stable diploids and haploids, and it's easy to make these constructs and to sporulate and to look at meiotic products. It's possible to transform it by integration and with plasmids, and even transform it with PCR products. You can also, that sounds like my granddaughter. Anyway, I'm sure some of you have got kids. It's also possible to have homologous recombination, and also to get simple targeting of mutations into the genome, and to do genetic screens. There's whole libraries of gene deletion mutations. Just a little bit of background. Someone might need to turn their microphone off. The yeast that I'm talking about in particular here is Saccharomyces cerevisiae. It's been used in genetic studies for over a year. We have libraries of yeast. Every gene has been deleted. There are about 6,000 genes. The genome's 12 megabases, so it's actually quite compact in terms of doing deletion studies. And many of the genes in humans have been identified based on their similarity in yeast. And of course, you can do the genetic studies, the knockouts in yeast, and find out what the genes do in terms of their coding, and you can complement many genes in yeast by complementation. For those interested in prions, yeast also have prions. They're safe, and you can use it to study prion diseases. And many similarities with human aging makes it a very valuable model for all sorts of things. So I've applied it to many areas over the years, and just using synthetic biology approaches. So the question that I wanted to go through here was, what does A-beta do to cells, and how can we design therapeutics that might be useful against for Alzheimer's disease? So we need to consider, as I mentioned at the start, one of the first characterisation of Alzheimer's disease was with plaques in the brains. So these plaques, of course, are extracellular, so we need to consider extracellular A-beta, but as well I've shown you that initially A-beta is produced inside the cells. So we need to consider both forms of A-beta and what damage they might do. So you can actually buy chemically synthesized A-beta from peptide synthesis companies all around the world. And what you find here is that if you put yeast cells in water, they're quite happy in water, and you can add whatever you want to them. If you add A-beta to them, you find that you can kill the cells if they're cells that have grown in an exponential phase. If they're cells that are in stationary phase, you actually stimulate their growth. This is an effect of the chemically synthesized A-beta. If you quantitate that, as shown in the next slide, you can actually do a dose response curve, and what you find here is we're looking at killing initially, so with relatively high doses you've got good killing, but at much lower doses you actually got this other effect, which I'm always interested in what happens at the lowest doses, not the really very high doses, but here you've got like say 500 nanomolar of A-beta, and it's causing a fourfold increase in cell numbers from that assay. So it's stimulating cells to divide. So I mentioned these cells are just in water, but in fact cells in water, they actually have reserves and they can actually go through these divisions. So those are the ones in stationary phase. They accumulate enough reserves so that they can go through those divisions. These ones may be triggered to do the same thing, but they're sort of growing a bit leaner, and maybe that triggering of growth could cause them to die. So that might be an explanation for what's going on there. But what's happening in Alzheimer's disease? Well, there are many papers that show a similar thing. With neuronal cells in culture, it actually kills the cells, and in brains, looks like it could do that by stimulating cell cycle re-entry. And of course, neuronal cells are terminally differentiated. So if you trigger them to go into a cell division cycle, it's deadly for them. So this could be what is happening in Alzheimer's disease. Okay, now I want to switch to producing native A-beta. This is producing A-beta inside the cells. What happens to them? Well, you can see here, these cells don't look very normal at all. I'll show you some normal yeast cells a bit later, but yeast are normally just a round cell, and you'll see a bud or an oval-shaped cell, and you'll see a bud coming off. They don't have these protrusions and odd shapes to them. So this A-beta is deleterious when the yeast is producing it. You've got, when you make a construct of yeast to produce A-beta, it grows slowly. Many cells can lose respiratory function, and that can be due to loss or rearrangement of their mitochondrial DNA. And you've also got this altered morphology, which can be seen under the microscope, and it can also be seen by looking at sizes on single cell sorters. Okay, if you look also staying with DAPI, so DAPI is a stain of DNA, and normally cells are impermeable, so cells can't be stained, but when I took this picture of cells in water with DAPI, I could see DNA leaking out of the cells. Okay, is there a question, or I'll keep going? Okay, we'll keep going. So the cells are permeabilised and they're leaky, and as well as that, they're sensitive to stress, to like heat. So here's an example of cells streaked out on a plate. This is a regular cell, a normal wild-type cell, and cells producing A beta. So here they are, streaked out. They're not growing too well here. This is their optimal growth temperature. And this is an identical plate or identical streak, and the plate's been incubated at the higher temperature, the maximum growth temperature, and you can see that they can't grow at that temperature. So they've got that increased sensitivity to heat. In addition to that, many of themselves can mutate and become resistant. And of course, once that's happened, we're not really studying the effect that we did want to study. So it makes looking at native A beta a bit tricky, but you can see some dramatic effects. So one of the things that we wanted to do was to visualise A beta more efficiently. So we made a fusion to A beta, and actually that settled down the toxicity a lot, and it helped the cells to grow a bit better when they were producing A beta, fused to jellyfish protein, green fluorescent protein. So these were constructs we made just by fusing A beta at the C terminus of a GFP or at the N terminus, and they were expressed in cells using plasmids like this, which are shuttle vectors that go into E. coli and into yeast. So these transformants, they produce GFP fused to A beta under the control of a constitutive promoter. So all of the cells are turned on to produce this protein. However, what you might notice here are only 20% of the cells fluoresce, and that's an important point I'll come back to. You can also do genome-wide screening and look at expression differences between these cells and these cells, and you can understand, you can picture all 6,000 genes on a microarray slide and work out what genes are upregulated and what are downregulated. And what you see is that there is a severe response to protein misfolding and oxidative stress, judged by what genes are turned on and up or down. Also, you'll note here that there's a difference in the expression. So GFP just spreads everywhere, but A beta GFP forms in these punctate patches in the cell. We haven't got the proof yet, but we think that is probably membrane blebs caused by the A beta GFP sloughing off membranes inside the cell. The cell can compensate for that, but that we think that could be what that appearance is. They're certainly novel structures. So I'm going to tell you a little bit about ageing in yeast, and so what happens with ageing is mother cells gradually get bigger, so you can tell an older cell by its size, and every time it has a young one, a bud, it leaves a bud scar which can be stained with chitin, which is, you can see staining here. And so here you can see a grandmother and two buds that she's had stained with chalcofluor. And here we're expressing GFP A beta, and you can see it's only in those cells. So the explanation for that, which I'll come to for that, is that what we're seeing is a very efficient turnover of A beta GFP in the young cells, but the old cells don't get rid of it. So this is the similarity between yeast and human cells, in that both exhibit a decline in protein, or thereabouts. Had the protein, it's just the old cells where you see the protein. They've all produced it, but just the old cells, the young cells get rid of it, the old cells don't. So this is actually means that we've got the basis of an assay for screening here. We can actually use this to perhaps look at compounds that might help these old cells to get rid of the unwanted protein. It could be a general aging assay, or we could use it specifically for Alzheimer's. So here's a comparison here. This is from a paper by, this paper looked at the hallmarks of aging, and I just want to highlight here the ones in green are those that yeast also exhibit with aging. So as I mentioned, we can tell the age of yeast cells by how many bud scars they have.
Young sir, I'm sorry to interrupting you, but your slide is not visible. Your slide, sir, I have shared the slide. Can you please give me the slide number at which you are?
I'm on 22.
Okay, sir. Okay, fine. Now I am just adding it.
Okay. Do you want me to stop sharing?
Yeah, I have started.
Okay. So on to slide 22.
Yes, so slide 22. Just a minute, sir.
Yep. So this slide just picks out a number of the hallmarks of aging in humans, and the ones in green here are also shared by yeast. And the particular one that I want to focus on is this loss of proteostasis, because I think this is why we have Alzheimer's disease and probably some other neurobiological diseases such as prion disease and Parkinson's disease as well, because we're not getting rid of unwanted proteins. Are we back on track?
No, sir, just a minute. I am just going to slide 22. So I have switched to slide 22.
Slide 22, yep.
Yes, sir.
I'll continue. So there are the hallmarks of aging, and as I was saying, I think the loss of proteostasis is a big factor. If we move on to 23, what is proteostasis, the loss of it? It's just like accumulating junk on your desk. That's going to lead to loss of your efficiency, and when it's in the brain, it's particularly disastrous, because the brain's a very sensitive organ and responsible for doing lots of important tasks. So you can lose function, you can have a build-up of other components that interact with the protein, possibly disabling them, and you can inhibit many processes as well. So I think this is one of the things that amyloid beta does. So how does it do that? Can move on to the next slide. So A beta can bind metals. In fact, it's found to associate with a number of metals: copper, zinc, iron, and some of these may drive cell proliferation. And of course, I've mentioned that's lethal to neurons. So we've actually done some experiments to look at that and see whether the A beta metal association is a factor. So what you can see here is we've put cells in water once again, and we've incubated them and then plated them out. So we've counted the number of colonies that are being plated before incubation and after incubation, equal aliquots. And all things, they're the same. But for transformants that have GFP-A beta, the number of cells was actually higher after incubation. So that's very interesting. It's those cells have an increased, they've got some division going on while they're sitting in the water. I'm going to move on to the next one. So we've actually looked to ask whether we can actually pick up changes for zinc and for iron, and that's shown on that slide there. So this is a bulk metal analysis. It's significantly higher for zinc and for iron. So the cells that the A beta that's fused to the GFP picks up the yep, you're right, slide 26. Yeah. With copper, there's no change, no significant change. And so we've actually asked the question, well, what if we just take wild-type cells and incubate them with metals? And you can repeat the same thing there. So zinc, we've titrated it. We've found out that zinc can actually stimulate cells to divide. Likewise with iron, and we've looked at the combination as well. So metals can cause increased proliferation. So this could be what A-beta is doing, recruiting metals and causing cells to proliferate in the brain and in these yeast experiments too. So what might we learn from that? Well, this means that we could perhaps try to selectively remove those metals from the A-beta, or we could try to remove the A-beta itself. Okay, now I'm going to switch tacks now and show you some experiments we've done using what we know from what's going on in people. So if we move to slide 29, there's a picture of statins. So these are some of the drugs that lower cholesterol. So these are widely prescribed drugs taken by many people. In fact, I've been taking simvastatin for 25 years. And there's of course many more, and there's more that are coming on the market because the patents for these have lapsed. They're outdated, and so they're generic now, so drug companies aren't making a lot of money out of selling these. But in fact, a study published back in 2007 showed that simvastatin looks to be the best drug for preventing AD. So there's a good take-home message today to tell, I know most of the audience is young, but you might have parents that are on statins. I'd suggest tell them to take simvastatin if they're prescribed a statin. And I'll show you the evidence in the next slide. So this was, Willowsen did a study of people in the Veterans Administration in the US. These are millions of people that have returned soldiers or been associated with the services. And they've looked at the incidence of Alzheimer's disease in those people over 3 years. And they know the prescribing information of them. And clearly you can see at this bottom left panel, those that are taking simvastatin have half the incidence of Alzheimer's disease. For Parkinson's disease, it's the same result for Parkinson's disease, but I'm only talking about Alzheimer's disease today. So they actually did other statins as well. So atorvastatin and simvastatin, these are the blockbuster drugs. The first statins to come onto the market, and these were selling $1 billion per year when they were first introduced. And they still sell a lot, but doctors will prescribe other ones quite often these days because of the pressure they get from pharmaceutical companies, I would tend to think. So moving down to the next slide, we're going to ask, what can we learn from yeast? Why is this the case? So these drugs are all designed to do the same thing, to reduce the cholesterol levels, and here you can see the pathway that they're working on. They're designed to reduce cholesterol, the endpoint in this pathway where statins are inhibiting HMG-CoA reductase, and they're actually having a bit of a broad effect. They're not just blocking cholesterol, but they're blocking other things, which include mitochondrial function. So some people feel a bit weaker with statins. You might be able to overcome that problem by taking coenzyme Q10 supplements. But importantly over here is there's an inhibition of protein prenylation. And I've mentioned that before. One of the proteases, base, is prenylated. And so if it's not prenylated, it doesn't cleave the APP to make A beta. So therefore, if you inhibit this pathway, you'd expect to have less A beta, and surely that's what happens. But it's only simvastatin that reduces the incidence of Alzheimer's disease, cutting it by half. So why is this the case? Well, it could be that, of course, we're not focused on cholesterol so much, but here necessarily, but we're looking at what gets into the brain. Simvastatin, and 20% of your cholesterol is in the brain. So simvastatin is the most lipophilic statin, so it probably gets into the brain and where it has its effect. But is it through cholesterol lowering? I tend to think it might be more than that, and we've done experiments to look at that. So I'll move on to the next slide. So this shows that in untreated cells, you've got, in this particular time, it was 12% that are fluorescing, and when we incubated the cells in the presence of 3 different statins, we got reductions in the number of fluorescent cells. So in other words, we're helping some of those old cells get rid of their GFP-A beta. And simvastatin does it best by far. So in yeast, we've shown that it helps in the clearance of the A beta. How it does that, we haven't actually fully addressed that yet, but it does it. It doesn't look to be through that regular pathway, but maybe an off-target effect. And if you look at the literature, you'll find that there are effects on autophagy and some of the other protein clearance mechanisms associated with other things. This suggests it's also happening with A beta. So this was GFP-A beta, and this, of course, is done by using a cell sorter where we look at the population of cells, and it's made particularly easy because the A beta is all targeted, tagged with GFP. But if we look at A beta and that constructs that produce the native A beta, you can show the same thing regarding native A beta. Here we don't look at single cells, but we look at the whole population, and you can see that simvastatin reduces native A beta by well over 90%. So it works there as well. So that's really good. This is the best drug for lowering the incidence of Alzheimer's disease. And for those you'll know many people that are on statins, and if you need to have a statin, I'd suggest try to get simvastatin. We'll move on to the next slide, it sums it up. And that will increase proteostasis in your life, leading to less A beta, well before you've got any hints of Alzheimer's disease. Can we take it further? Because, not everyone gets prescribed or needs to be on a statin to lower cholesterol. So using this assay, we can actually look at other things too. We can look at things that improve proteostasis, and we can look at things perhaps that reduce proteostasis and find out things to avoid. So if we move on to the next slide, I'll just show you some other applications where we've used A beta. So here, as I said earlier in the lecture, some Alzheimer's is sporadic, so it's not always clear what's going on. And aluminium is sometimes being linked with Alzheimer's, and there's a lot of controversy surrounding aluminium. So we actually did this study and showed that aluminium plus A beta leads to much less growth. So there's an inhibition, there's some toxicity associated with that, as you can see. If we look at cells producing just GFP versus those producing GFP fused to A beta, A beta causes a more detrimental effect in the presence of aluminium. Another compound, a tyramine, which is found in foods, there's not much in the brain, but it's also a toxic component. So we've toxic synergy there. So we've looked at quite a few things and some have no effect, but these two did have effects, and there's a lot more searching that we could do to take that further. I'll just move on to the second last slide. So things that help. So polyphenols, there's a strong association between polyphenols, things like curcumin and turmeric and green tea and so forth. We've been able to screen lots of those compounds in this yeast assay to see what effects they have on reducing the percent of cells that have green fluorescence. And some of the beneficial effects of these are known from neuronal cell studies as well. And we've actually looked at patients. But with the yeast, we've been able to do that. So here you can see we've picked up on trans-chalcone and bicolin, and together they have a very synergistic effect in reducing the amount of amyloid beta in cells. So we've only just published that study last week to show that. So the last slide, I guess, is just to go back over the students and the teachers who've contributed to these studies at RMIT University, and to thank them for all this work. We certainly value what teachers do, and we value the enthusiasm of students as they pick up on some of these things and take it forward and become teachers themselves. So I'll finish there, and happy to take questions if you've got any.
Thank you so much, sir. Thank you so much for enlightening a lot of knowledge and your experience. Now I will request the participant, if you have any question, you can ask. Participant, if you have any question, any query, you can ask.
There were a lot of hands up at the start.
Is there any query?
Yes, sir.
Yeah.
I have a query, sir. Sir, my father's when corona pandemic is very in a high peak in the month of May in India. So my father is suffering from pre-symptoms of coronavirus, and so I want to ask a question that if anyone can see this predefined symptoms of this Alzheimer's disease after the coronavirus, what are the symptoms of this disease that one can find in your family?
Well, there's memory loss, brain fogginess. So coronavirus causes a lot of brain fogginess and so forth, and there's this thing called long COVID, where that may persist for a long period. We don't know how long it might persist for, but there's certainly a lot of research going on in that at the moment, and things like fibromyalgia and chronic fatigue will probably get a boost from the coronavirus studies, because similar things happen there, and now we're sort of realising just the connection between these viruses and what they do. Yeah, there's probably, I don't think coronavirus can add all sorts of complications if there's comorbidities. I don't know whether it can bring Alzheimer's on earlier, but there can sometimes be cognitive defects that don't resolve, that are temporary. Alzheimer's tends to be something that's just progressive and doesn't subside. There doesn't appear to be a real cure, so I think the hope is that we can prevent it rather than cure it. So that's why it's worth taking care of yourself in the younger years, right before there are any symptoms.
Okay, thank you, sir. Thank you so much.
Is there any other question? Okay, I think we are. Yeah, okay.
Sir, one of your slides shows the fluorescent intensity of a cell. Sir, that fluorescent was measured by using any model system?
So we have the jellyfish green fluorescent protein used for A beta. So it's tagged. We can see where it is, and we can monitor what it's doing. And you've got those punctate patches that you see. Yeah, GFP is all sorts of proteins. It's a good marker for tagging.
Sir, Alzheimer's is the accumulation of amyloid beta, and the slide shows that the level of accumulation is decreasing. So we are taking the model, a mutant model, a strain which is tagged with that amyloid beta or anything else.
The A beta accumulates today, not decreases, it accumulates, it increases. And sorry, I missed the last part of the question. It's gone from me. The Icelandic mutation that results in 40% less A beta. Have I answered the question, or just ask it again if you like?
Sir, if possible, please do provide us email ID so that we can post our questions. Due to some network issue, we are not able to ask the question. My question was very simple, sir, that you have used a model which shows that the accumulation of amyloid beta has decreased to 50%. Sir, just I want to know which model was that, and was that model was a mutant or a strain?
Okay, so the assay that we have is based on a population of cells. So it's looking at the whole population. About 25% of the population is old and has that green fluorescence. So the old cells are marked by not being able to turn over the protein. So we use a flow cytometer. I guess you could use a microscope, but it would be very laborious. So we use a flow cytometer and we count the maximal green cells, and we treat the cells. We do various treatments, so we have untreated versus treated cells. So we're constantly looking at the percent of green fluorescent cells. There are other stains that we can use as well, such as dyes to measure how much oxidative stress there is.
Actually, I am using an animal model for the study of Alzheimer and Parkinson, and I used mutant strain which is tagged with amyloid beta, and I use my different molecules extract on that, and we go through the intensity through fluorescent microscope. So that was my only.
So people with animal models of Alzheimer's, they often express the human APP in mice, and they overexpress the proteases. So they might have 5 copies of the human gene. They overexpress everything, and I'm not sure whether it's always legitimate, because you've got higher levels of APP, higher levels of the proteases, so it's artificial. We're able to produce our proteins as in the cytoplasmic form, or the A beta, a native A beta is secreted. We have a leader sequence that takes it through the normal pathway of the endoplasmic reticulum. The leader is cleaved. What's produced is native A beta, which we can see on the mass spectrometer and measure levels that way.
Thank you, sir.
Thank you so much, sir. I think we all are very much satisfied and enlightened on the search on Alzheimer and Yeast and the role of Yeast. Now, I think if you have any query, we can take it. Otherwise, I will transfer it to Organizing Secretary, Dr. Rajesh Srivastava, for a note of thanks. Over to Rasi Srivastava.
Thank you, Dr. Sapna sir. Namaskar and good afternoon to all.
Thank you. And my email is just my name, ian.macreadie@gmail.com if you do have questions. It's not spelled correctly in that caption, but you know the correct spelling. Okay. Thank you, everyone. Thank you. Thank you.
Thank you, Dr. Sapna sir, for giving me the privilege to offer the vote of thanks in this international webinar organized by School of Biotechnology, IFTM University, situated at Moradabad, the brass city of India. Initially, I must say thanks to Almighty. Now, before moving to my vote of thanks, I must congratulate everyone on the occasion of Teachers' Day, which is being celebrated across the country to the mark of birthday of Dr. Sarvepalli Radhakrishnan, former President of India and Bharat Ratna awardee. On the behalf of IFTM family and School of Biotechnology, I, Dr. Rashi Srivastava, Organizing Secretary of this webinar, extend my heartfelt gratitude to the distinguished speaker, Sir Macreadie, whose brief profile has been read out by Sapna sir, which is clearly shown us his contribution in the field of biotechnology. I am thankful to Jan sir for enlightening all of us how Yeast model for Alzheimer disease can contribute to the development of treatment and preventative strategies for Alzheimer disease. I am confident that the talk has been great knowledgeable experience for all of us. Once again, thanks to Jan sir for sparing his valuable time for his busy schedule. I owe to the respected Chancellor Shri Rajiv Kothiwal ji, respected Pro-Chancellor Shri Abhinav Kothiwal ji, respected Vice Chancellor Professor M.P. Pandey ji, respected Registrar Professor Sanjeev Agarwal ji, who have given the opportunity to organize this international webinar on the occasion of Teachers' Day. Thank you, sir. My sincere thanks and gratitude to all the audience and alumni for joining this webinar and patience hearing. I also thanks to all of them who got registered but could not attend the webinar. I am grateful to the Director Sir Dr. Tanzeel Ahmed as Chairman, Head Sir Dr. Nabil Ahmed as Co-Chairman and Dr. Sapna Sir as a Convener. Without their support, this webinar would not have been possible. I also submit my thanks Shri K.K. Bansal Sir, who inspired us to organize this international webinar. I appreciate the team of department, Mrs. Shweta Srivastava, Mrs. Ritika Saxena, Mrs. Kanchan Lakhera, Mrs. Kriti Sheenet, Ms. Shweta Singh, Mr. Utkarsh Tyagi and Mr. Ankit Bhardwaj for everything done by them. My vote of thanks will be incomplete if failed to thank the technical team, non-technical staff and all other involved. So I am thankful to technical team, non-technical teaching staff and all involved directly and indirectly in organizing this international webinar. Once again, thanks to Almighty and dhanyavad to all. Over to Dr. Sapna sir.
Thank you, Rasi ma'am. I think we are very blessed that we are working IFTM University, where everyone is very supportive in terms of technical team who are always ready to support us. And I think this lecture was really very informative for us. And we have learned a lot. Thanks to everyone who are joining the webinar. And at the end, the e-certificate will be provided to your registered email ID. With this word, I am thankful to all of you. Thank you, sir. Thank you, sir. Now I'm going to just close the session. Thank you.
Thank you, everyone.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir. Thank you, sir.
Thank you.
Thank you so much, sir.
Bye-bye, sir.
Thanks, sir. Happy Teachers' Day.
You're welcome. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
That is
Thank